Hello everyone. Let's talk about chromatin remodeling today. I hope you remember we discussed previously that genes can be regulated at different stages, and whenever we want a gene to get expressed, the first thing we need to do is to relax the chromatin. This is where modification of the chromatin takes place, and we need to relax the DNA so that it becomes accessible for the transcriptional machinery. And then there are all other steps involved in the regulation of gene expression, which include transcription, post-transcriptional modifications, and then regulation at the level of translation, and even after the synthesis of the protein, it may be regulated, whether it is needed or not, depending upon the situation. However, the first thing that we need to do whenever we want a gene to get expressed is to relax the chromatin, which is present in a compact form. Today we will talk about the different phenomena, how this chromatin can be relaxed. So let's move to our slide for chromatin remodeling. Yeah, so we would be basically talking about two phenomena: histone acetylation and histone methylation. First we will talk about the acetylation, and then maybe in the next lecture we'll talk about methylation. But anyhow, first of all, we need to look at the structure of what we call nucleosomes. What are nucleosomes? Nucleosomes are composed of four different types of proteins, which are named as H2A, H2B, H3, and H4. So these four proteins are present in pairs. It means this disc-like structure contains a total of 8 molecules. 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3, and 2 molecules of H4. So putting together, this constitutes an octamer. This constitutes an octamer, which is known as a histone disc, or a histone core. So this disc or core is the function of these histone proteins is to provide stability to the DNA. So what happens that the DNA goes 2 turns around this octamer of histone proteins, and then there's a short strand of DNA which is not associated with any histone proteins, and then it goes another 2 rounds around another disc, and then so on. So this segment, which is composed of the histone proteins plus the DNA, this structure is called as nucleosome. So these nucleosomes give stability to the DNA. So if we use an enzyme like a nuclease enzyme, so it would be easier for the nuclease enzyme to digest the linker DNA, because it is now not associated with any proteins, and the DNA which is associated with histone proteins is comparatively more stable, and it would be more difficult to digest this DNA. So we can fine-tune the amount of nuclease enzyme that is needed to digest the DNA, and this way we can release these beads or these nucleosomes from this string. So this string is approximately 11 nanometer in width, where 2 nanometers is the width of the DNA, and then approximately 10 nanometers is the width of these histone proteins. So putting them together, this bead string gives us a width of 11 nanometers. And then these beads on a string, they're put together into a primary or a first tertiary structure, which is called as a 30 nanometer chromatin fiber. So what we are doing, basically, we are moving upwards in the picture in a backward direction. What we are trying to do is to condense the chromatin. So in order to condense the chromatin or the DNA into the form of a chromosome, what we need to do, we need to put the DNA onto these histone proteins so that this string can be constituted. And then we put together multiple of these strings 2 or 3 of these strings. They are packed together, which gives us approximately 30 nanometer width chromatin fiber. So this is the first tertiary structure of the chromatin. And then this 30 nanometer chromatin fiber is associated with some non-histone proteins which constitute the scaffold, where this 30 nanometer chromatin fiber is associated. So these are non-histone proteins. These are called as scaffold proteins. They help in chromosome condensation to ensure the fidelity of chromosomes during cell division, and they also constitute they are the backbone structure after we deplete the histone proteins. So this backbone structure is positioned along the chromosome axis, hence called the chromosome scaffolds. So these scaffold proteins are basically providing stability to this 30 nanometer chromatin fiber. So this chromatin fiber, along with this, this 30 nanometer fiber, which is associated with these scaffold proteins, these are looped into, once again, so the width of this structure, the scaffold proteins plus the 30 nanometer fiber is approximately 300 nanometers. And then these scaffold proteins and this nucleosomes fiber, they're further condensed together into a 700 nanometer structure, which is approximately the width of one chromatid of a chromosome. So if we go the other way around, so we have a chromosome in the metaphase in a compact form. So this is the chromatid. If we enlarge the chromatid, the chromatid consists of, so this is approximately the width of the chromosome is 1400 nanometers, one chromatid 700 nanometer. And if we take this chromatid, we see that this chromatid is composed of scaffold proteins which are associated with the 30 nanometer chromatin fiber. So where each, the width of this scaffold proteins is approximately, along with the 30 nanometer fiber, is approximately 300 nanometers, and the width of this 30 nanometer fiber is obviously 30 nanometers, and the width of one string of nucleosomes is approximately 11 nanometers, and the DNA wrapped on it is 2 nanometers. So the DNA which is wrapped around these nucleosomes is approximately 147 base pairs, and it goes slightly less than 2 turns around one disk, and the length of the linker DNA in between, that varies with the species, and it ranges between 15 and 55 base pairs depending upon the species involved. So here's the 30 nanometer chromatin fiber. This is also known as solenoid model. Solenoid is a cylindrical coil of wire which acts as a magnet when it's carrying current. So this is where the name comes from. So as you can see, these are 30 nanometer condensed chromatin fibers, and each single fiber is composed of these 4 different types of histone proteins. So 2 molecules from each protein, H2A, H2B, H3, and H4, and in total there are 8 molecules. And the DNA is going 2 times around this histone octamer. So and in between there's a linker DNA, and as we can see, there's also a 5th type of histone protein. So there are 4 types of histone proteins inside this histone core: H2A, H2B, H3, and H4. And here's a 5th type of histone protein, which is known as H1 protein. This H1 protein is not part of the nucleosome. It's rather associated with the linker DNA. So basically, this gives H1 histone protein. This gives stability to the chromatin structure. This is not part of the nucleosomes, but this gives stability to this structure, to this string. So let's proceed further. So what we see here on the left is the space filling model of a nucleosome. This is the lateral view and this is the front view. And on the right we see this is the ribbon diagram. So let's see what do we have here. We see this core of proteins, which is composed of 4 different types of proteins, each indicated via a different color. So yellow color is one type of protein. This is H2A. So red is H2B, and blue is H3, and green is H4. So there are 4 molecules of each protein, 4 on this side and 4 on the other side of the nucleosome. And what you see here, this fiber-like structure, this is the sugar phosphate backbone of the DNA, or this is the DNA which is going 2 times around this nucleosome, and which is clearly visible in this lateral structure. So this is going around the histone octamer 1 time, goes to the other side of the histone proteins, comes back, and then wraps around it once again. So if we look at the ribbon structure, we see that in the middle we have got these 4 histone proteins, H2A, H2B, H3, and H4. But what else we see? We also see some dotted lines coming out of this structure. So basically, these dotted lines indicate what we call histone tails. These histone tails are not part of the core structure. So usually they are located at the end terminus, and these are composed of roughly 11 to 37 nanometer, 11 to 37 amino acids, and usually they are located at the end terminal. But H2A also has 2 tails at the C terminal, and all of these histone tails, they're coming out of this core structure of the nucleosomes. So it means they would be visible out of this nucleosome, and they will penetrate through these grooves of the DNA and then come out of the DNA towards the periphery. And these histone tails have the ability to interact with the linker DNA or with the neighboring nucleosomes. So if we go back here, so this is the octamer of the histone proteins. That should be better visible on another slide. Let's see here. So this is the octamer of the histone proteins. This is the DNA, the blue line or the blue thread going around the histone proteins, 2 turns around each octamer. And these thread-like structures, these are basically the histone tails, which are coming out of these histone proteins. And these tails have, because they are composed of different amino acids, they are supposed to carry a charge. So generally the lysine residues or the lysine amino acids on these histone proteins, they have positively charged side chains, and we know that because of the presence of the phosphate groups on the DNA carries a negative charge. So these histone tails, whenever they're positively charged, they can interact with this linker DNA, and they can pull the DNA towards themselves, and pulling of the DNA and the neighboring nucleosome, so they can interact with both of them. They can interact with the DNA, and they can also interact with the neighboring nucleosome, and this interaction leads to the compaction of this chromatin. So the nucleosomes come together and they become condensed because of the oppositely charged molecules on the histone tails and on the DNA. So the phosphate gives negative charge to the DNA, and lysine gives a positive charge to the histone tails. So how do these histone tails get acetylated? Is because of different enzymes. We call them writers. These writer molecules can carry acetyl groups from another molecule and then add them onto the lysine residues, or even sometimes arginine residues on the histone tails. And what happens, that addition of acetyl group masks the positive charge on the tails. And when the positive charge is masked on the tails, then this positive charge cannot interact with the negative charge on the DNA, and this leads to the relaxation of the chromatin. So in this case, in compact form, the genes are turned off. So if you want a gene to get expressed, you need to relax the chromatin. And in order to relax the chromatin, you need certain enzymes which can write this code on the histone tails, which can add something onto the histone tails. And these writers are generally known as histone acetyl transferases. So these histone acetyl transferases can transfer acetyl enzyme from acetyl coenzyme A and then add it on to the lysine residues in order to mask their positive charge. While there are also other enzymes, so that would lead to the relaxation of the chromatin, because the tails would not then be interacting with the DNA, and these nucleosomes will move away from each other, and that would lead to the relaxation of the chromatin and then ultimately turning on of the transcriptional activity. On the other hand, we also have some other enzymes, what we call erasers. These are generally histone deacetylases, and the job of these enzymes is to remove this acetyl group. So the removal of acetyl group from the histone tails would leave a positive charge on the histone tails because of the presence of the lysine residues, and this positive charge will then interact with the negatively charged DNA and then pull it towards itself, and this pulling leads to the compaction of the chromatin and ultimately switching off of the gene. We will talk about the readers later. So at the moment we are talking about two things. Acetylation leads to the relaxation of the DNA, and deacetylation leads to the compaction of the DNA. So mostly acetylation takes place on H3 and H4 histone tails, but the largest or the greatest effect is achieved by the acetylation of H2B and H4 histone tails. Let's see how does it take place on the how does this acetylation take place on the lysine residues. So here's the side chain of lysine amino acid. So we see there are two amino groups, one at the alpha position and the second one at the epsilon position. So this is the alpha, beta, gamma, sigma, and epsilon position. So there's an epsilon amino group and there's an alpha amino group. So this is the epsilon amino group, which basically gets masked by the acetyl group. So what do histone acetyl transferases do? They will carry an acetyl molecule from acetyl coenzyme A and add it on to this amino group. And addition of this acetyl group would mask that positive charge. So now the lysine residues are not positively charged anymore, and these tails cannot interact with the neighboring DNA, hence the chromatin is relaxed. While on the other hand, if we talk about the histone deacetylases, what does deacetylation do? These enzymes would remove the acetyl group and leave the lysine residues positively charged because of the presence of these amino groups. And then this positive charge will interact with the negative charge of the DNA and pull the nucleosomes together. So here we see this thing again. So acetylation leads to the masking of the positive charge on the lysine residues, and this leads to the relaxation of the chromatin. While if you deacetylase, if you use deacetylase enzymes, so what they do, they would remove the acetyl group and would leave the lysine residues or the lysine amino acids on these tails positively charged because of the amino group on them, and this positive charge would then attract the negative charge of the DNA towards itself, and this would lead to the compaction of the DNA. So by the addition of acetyl group, you can relax the chromatin, and by the removal of the acetyl group, you can compact the chromatin. So in addition to that, it's not just acetylation and deacetylation that takes place on the chromatin. Methylation can also take place on the histone tails, and in addition, certain other amino acids like serine and threonine, they can also get phosphorylated. So phosphorylation would introduce a negative charge. So phosphorylation would also lead to the relaxation of the chromatin. In addition to that, sometimes these histone tails can also be tagged with a single ubiquitin molecule. This ubiquitin is it's a 76 amino acid molecule. I hope you remember from your cell biology class that the addition of multiple ubiquitin molecules could target the proteins to proteosomes for degradation. But this is different in this case. It's not addition of the multiple ubiquitin molecules to the histone tails. It's rather one ubiquitin molecule that can be added to the histone tails. And the addition of this ubiquitin molecule does not affect the stability of the histones. It rather influences the chromatin structure. So I hope you could understand what is how does chromatin get relaxed and how can we condense the chromatin. So you understand how does acetylation take place and deacetylation take place on the chromatin and what is its impact. So we will talk about the methylation in the next class. Thank you very much.